So for tonight's lecture, we're going to go over kind of the ending of part C of chapter 3. So remember there's 4 PowerPoints in chapter 3. It's really long. You are responsible for all the information, but I'm kind of skipping over the parts I'm hoping you guys had in anatomy. The first part of part C of this PowerPoint is all on cell organelles in the cytoplasm, like the mitochondria. And then I'm going to end here talking about the nucleus because we're really going to focus on transcription and translation and creating protein from DNA tonight. So we're going to start with the nucleus because this is kind of where DNA is stored. The nucleus is the largest organelle in the body, in the cell. An organelle means tiny organ. The nucleus contains all the genetic information. We often call it the blueprints. So your nucleus contains your DNA, which your DNA is just the blueprint for all of your cellular proteins. Most cells have one nucleus, but some can have many nucleus. Some skeletal muscle, certain bone cells, and some liver cells. And the only one that does not have a nucleus, the only cell, is the red blood cell. So the structure of the nucleus, it has a nuclear envelope, a nucleoli, some nucleoli throughout it, and chromatin. So here's a look at the nuclear envelope, which surrounds the nucleus, and it has nuclear pores going through it. Chromatin will be condensed DNA, so the genetic blueprint. And then the nucleolus is the central part of the nucleus there. The nuclear envelope is just the double membrane barrier that encloses the jelly-like fluid, the nucleoplasm. The outer layer is continuous with rough ER, and that's because your endoplasmic reticulum will be studded with ribosomes, the rough part of it will, and that will help with protein synthesis. And the nuclear pores just allow substances to pass in and out of the nucleus. So here's a look at the nucleus and the nuclear pore complexes, the nuclear lamina, which is the inside of it that helps with the structural support. Nucleoli are dark staining bodies that are circular and shaped within the nucleus that are involved in ribosomal RNA synthesis. And we'll talk about what that is really next part of chapter 3. And there's usually only 1 or 2 per cell. The chromatin consists of 30% thread-like strands of DNA and 60% histone proteins and about 10% RNA. It's arranged in fundamental units called nucleosomes, which consist of DNA wrapped around histones. And histones are just proteins that DNA will wrap themselves around. During a part of mitosis, the chromatin will form chromosomes, and chromosomes are just condensed chromatin, where we get DNA wrapped very tightly around those histones, eventually forming chromosomes. And chromatin forms your chromosomes right before your cell divides. So this is the structural look of DNA. It's this spherical double helix structure. We'll talk about in a little bit what makes up the helical shape to DNA. And DNA winds around itself really tightly around these histone proteins, which kind of look like tablets. And then they're wound extremely tightly eventually until they make chromosomes during cellular division in mitosis. So here's some more chromosome structure. Eventually they'll become extremely tightly wound that they'll make this X-like structure, which is what your chromosome looks like. All right, so that was the end of this part C of chapter 3. And I want to continue with the final part of this chapter, part D. Can everyone see the part D here? This is hopefully better. All right, so hopefully part D, chapter 3 is in front of you now. And we're going to start with the cell cycle, which is the series of changes that the cell undergoes from the time it's formed until it reproduces. There's 2 main periods in the cell cycle: interphase, where the cells grow and carries on its normal activities. So if it's a muscle cell, it continues to contract. If it's a cardiac cell, it will continue to form its normal functions. In cell division, then, is the mitotic phase. And the mitotic phase is when the cell divides into 2 identical daughter cells through mitosis. So interphase is the period from cell division, cell formation to cell division. And this will just be routine activities. The cell spends about 90% of its time in interphase, just normal processes. During interphase, the nuclear material will be uncondensed in the chromatin state. It'll consist of subphases in which DNA will be replicated. And these are the subphases, G1, S, and G2. G1 is a growth phase. Cells that stop dividing are said to be in G0 phase, which is even before this. S is a synthetic stage, and the important part about S phase is that this is where the DNA replication occurs. And we need to replicate the DNA into two identical strands so that when the cell divides, it gets that identical piece of DNA into the daughter cells. And that's why we say in mitosis, we form two identical, genetic identical daughter cells. And G2 is the preparation for cellular division. So if we look at the cell cycle in a circle, we see our G1, S, and G2 phase. There's a little checkpoint that restricts growth at a certain point that we'll talk about where that comes into play. And then we get to our mitotic phases, which are our PMAT, which I call them, if you've had me before, prophase, metaphase, anaphase, and telophase. And then cytokinesis is not technically part of mitosis, that's just the eventual cell splitting. So again, the majority of the times, all of your body's cells are in the normal interphase stages where they're just growing, replicating their DNA. They're performing out their normal cellular functions of creating ATP or digesting bacteria or fighting off viruses. And so the majority of the time, they're just doing that normal function stuff. But about 10% of the time, 10 to 15% of the time, they will be in the division or mitotic phase. So DNA replication, this is when we replicate our DNA. The double-stranded DNA helical molecule will unwind and unzip. It uses a replication fork, where's the point where the strands will separate. You guys don't need to know all the details, but DNA replication goes or happens in interphase. DNA polymerase is just the name of an enzyme that attaches to the starting kind of primer protein and begins to add nucleotides to form a new strand. So DNA polymerase will synthesize both new strands at the same time, kind of one leading and one lagging behind it. All right, the end result, again, in interphase, we get two identical daughter DNA molecules. They are identical to the original copy of DNA. And then during mitotic cellular division, one complete copy of that DNA will go to one cell and the other complete copy will go to another daughter cell. We call this semi-conservative replication because we start with one strand of DNA, it splits, and then the daughter strands of DNA are replicated on the one half of the original strands. So here's a look at replication of DNA, just a summary. So here's our chromosome, here's our old DNA, and enzymes will attach to the old DNA and split it apart, and we call the splitting of the original old DNA the replication fork. So the DNA will split at that replication fork, and then a leading and a lagging strand, these are just the names of the new DNA strands that will be formed with one half of the old DNA strand. So again, at the end, we get two new strands of DNA that are genetically identical to the old DNA. And that's possible because of these base pairs. So where my cursor is, you have base pairs, adenine, thymine, cytosine, and guanine. These are base pairs of nucleotides, and they will only bind with certain pairs. So adenine and thymine always bind together, and cytosine and guanine always bind together. So when we separate out our old DNA strand, a new DNA strand will bind with the correct base pairs to create a replicated identical copy of the old one. So this is just DNA replication. We need it for it to happen before we divide the cell because we need to put a complete new strand of DNA in our daughter cells. I'd encourage you guys to watch this video, replication of DNA, on your own. Again, you'll have to download the PowerPoint to your device to actually see that. Cellular division then is mitosis. Your cells need to replicate themselves continuously for growth and repair, especially your skeletal, cardiac, and nerve cells, they do not divide very efficiently, so damaged cells will be replaced with scar tissue. Mitosis is divided into, the mitotic phase is divided into mitosis and cytokinesis. And control of cell division is extremely crucial, so cells divide when necessary, but do not divide unnecessarily. And this is the case in cancer. Cancer cells divide without stopping, so that's why it's extremely difficult to kill off cancer cells, because they never stop dividing. It's uncontrolled cell growth is the literal definition. So now we're going to get into the mitotic phases here, starting with the PMAT phases and then cytokinesis. So mitosis is the division of the nucleus in which that duplicated DNA will be distributed to two identical daughter cells. These are the stages: PMAT, prophase, metaphase, anaphase, and telophase. You should know the difference between early prophase and late prophase. That is it's an exam question. So in early prophase, the chromatin condenses, forming chromosomes. Remember, before, when we're not in mitosis, your DNA is just hanging out as chromatin. But in early prophase, the chromatin starts to condense to form chromosomes, which will be easily pulled apart later on. Each half of a chromosome is called a sister chromatid, and they're held together in the center by a centromere. We got a lot of C words here. And a centrosome and its duplicate begins synthesizing microtubules that push each centrosome to opposite poles. In late prophase, the nuclear envelope breaks up. So again, the DNA at this point is still in the nucleus. So early prophase, the DNA forms chromosomes, and in late prophase, the nuclear envelope breaks up. This is important because eventually we want to separate out the chromosomes that are in the nucleus. So we got to get rid of the barrier of the nucleus itself. So here is a look at interphase showing normal cell growth. So during interphase, you would see chromatin, you would not see chromosomes, and you would see the nuclear envelope still intact. Here's early prophase where we would start to see chromosomes forming. And here is late prophase where we would start to see the division of the nuclear envelope. So the division of the nuclear envelope and the chromosomes are still in their X shape, but they haven't been lined up or split apart yet. Metaphase is when the centromeres of the chromosomes line up at the cell's equator, so right down the middle of the cell. So we call this the metaphase plate, or the equatorial plate. So the chromosomes line up in metaphase. You can see those there. We start to see what we call the spindle form, which are spindle fibers that are reaching out and connecting to the centromeres of those chromosomes. Then anaphase, this is the shortest of all phases. This is just when the centromeres of the chromosomes are split or pulled apart. And I'll show you a picture of that here. So in anaphase, the chromosomes are now being split apart into their sister chromatid halves. And telophase is begins when the chromosome movement stops. So a new nuclear membrane will start to form around each chromatin mass. So at this point, the chromosomes will start to uncoil and go back to chromatin. The spindle will disappear. And in cytokinesis, this is when the two daughter cells will be completely pinched apart. So here we have kind of telophase and cytokinesis similarly, not at the same time, but telophase, the nuclear envelope will start to reform. The chromosomes will start to uncoil back into chromatin. And in cytokinesis, we'll see a new cell wall forming between the two cells. Cytokinesis means kind of cutting up of the cell. So that's what happens in cytokinesis. And this is a great animation that you can watch with that too. Questions so far on any of this? Okay, cell division. In order to control cell division, because we don't want your cells to divide completely continuously. Thanks, we have different start and stop signals, which help help to direct when a cell should and should stop dividing. Go signals include critical surface to volume ratio. So this just means if the cell needs more space, it'll divide. Other chemicals could be a go signal, like a growth factor, which is telling the cell to grow and divide, or other hormones. Stop signals might include availability of space, so normal cells will stop dividing if they come into contact with other cells of the same type. This is referred to as contact inhibition. More groups of proteins that are crucial to the cell's ability to accomplish the S phase, which is that DNA replication phase. Cyclins, CDKs, cyclins. These are just more proteins that accumulate during the interphase and will activate the S phase, which is DNA replication. Checkpoints are also key. So we have a G1 checkpoint, which is the restriction checkpoint, which is the most important. If a cell does not pass this checkpoint, it'll enter G0, in which no further division occurs. This will happen if the cell is some sort of faulty, or if there's something wrong with the cellular DNA. It won't get past this checkpoint until the repairs in that DNA are made, and it might not get past that checkpoint at all. So it's kind of your body's way of not passing on something genetically wrong to a future daughter cell. So again, here's the cycle, and this was that restriction checkpoint that happens around G1. Here's another checkpoint, but the G1 checkpoint is probably the most important. Then we get to protein synthesis, and this is kind of where I wanted to spend the majority of the time. So protein synthesis is just when we turn the genetic blueprint in your DNA into a protein. And when we talk about proteins, or we talk about genetic material to create proteins, we're really talking about everything in your body, because every thing in your body is made of these proteins. So your genetic blueprint in your DNA is for all of the proteins in your body, but that includes all of your traits, what eye color you have, how many fingers and toes you have. This is all in the genetic blueprint. The DNA will direct the order of amino acids in a polypeptide. Polypeptide is just another word for a protein, and proteins are made of 20 different amino acids, but different combinations of them. A segment of DNA that holds the code for one polypeptide or protein is referred to as a gene. And the code is determined by the specific order of the nitrogen bases in the DNA, or in that DNA. And these are the bases: adenine, guanine, thymine, and cytosine in DNA. And the code will consist of 3 sequential bases, and we call that a triplet or a codon, where a GGC base pair will code for the amino acid proline, and GCC will code for arginine. So this kind of triplet or codon of 3 base pairs will code for a specific amino acid, and then we take each triplet and just start putting amino acids next to each other to create a protein. Genes are composed of what we call exons and introns. Exons are part of the gene that actually codes for the amino acid, and introns are non-coding segments. So introns will be taken out of the gene, and we're just left with exons, which will actually be the coding portion for the genetic amino acids. This is an animation that describes the differences between DNA and RNA, and I'm going to tell you right now what they are, but you can watch this on your own as kind of a summary of that. You'll notice right off the bat, DNA is double-stranded and RNA is single-stranded. So what is RNA? Your RNA, ribosome nucleic acid, is the go-between molecule that links DNA to proteins. So in order to get from DNA, your genetic code, to eventually a protein, we need something in between. And this is where RNA comes in. RNA is ribose nucleic acid. I said that wrong, excuse me. All RNA is formed in the nucleus from DNA, and RNA differs from DNA in a couple ways. RNA does not have the base pair thymine in it. It substitutes thymine for uracil. So it'll have adenine, guanine, cytosine, and it'll have uracil. RNA has the ribose sugar instead of the deoxyribose sugar, and that's where we get its name. DNA is deoxyribose nucleic acid, and RNA is ribose nucleic acid. And there's 3 types of RNA, and they each have a specific function in creating protein from the DNA. Messenger RNA, ribosomal RNA, and transfer RNA. So again, we're looking at the roles of RNA, and RNA will be the go-between that links your DNA to your, eventually gets a protein out of it. So let's start with messenger RNA. This is single-stranded. Messenger RNA is a code from a DNA template strand, and it's copied with complementary base pairs, resulting in a strand of messenger RNA. And this process is referred to as transcription. So transcription is just taking DNA and creating an RNA copy of that DNA. So we know that DNA has the genetic blueprint for proteins. Well, in order to turn that genetic blueprint into RNA, we have to go through a process called transcription. Your messenger RNA will maintain the same triplet code or codon from DNA that had 3 base pairs for every amino acid. So that's what messenger RNA is, and I'll talk about how it all kind of comes together. Ribosomal RNA is the structural component of ribosomes. So maybe you've learned or can remember that ribosomes will create proteins, and now you're going to find out how that occurs. Ribosomal RNA is just the structural component of ribosomes. So our RNA just is in ribosomes to help make proteins. Along with transfer RNA, it also helps to translate the message from the messenger RNA into a protein polypeptide chain. And then your transfer RNA is called transfer RNA because it will actually carry the amino acids to a specific codon sequence on the messenger RNA. So the transfer RNA has special areas that contain a specific triplet code that will allow the transfer RNA to carry only a specific amino acid. And that transfer RNA base pair will be complementary to the messenger RNA. And this is when what we call translation, when we actually are starting to join the amino acids together. So then how does this all come together? How do we make proteins from DNA? It occurs in just 2 steps, 2 simple steps. Transcription is the first step. And transcription is when we take our DNA information and we turn that into a messenger RNA strand. So we go from a double-sided, double-stranded DNA molecule to a single-stranded mRNA molecule, and that's called transcription. This happens inside the nucleus. And then in translation, messenger RNA leaves the nucleus and it goes to ribosomes, where the ribosomal RNA will help to assemble polypeptides when a transfer RNA brings along amino acids. So here is kind of a general summary of where DNA protein synthesis occurs. Here's the nucleus, and transcription occurs in the nucleus. And again, transcription is just taking DNA and turning the genetic code into a single-stranded mRNA molecule. This is a pre-mRNA and a mature RNA, but transcription just occurs when we take DNA and we form it into an mRNA molecule. If it helps you remember, mRNA stands for messenger RNA because it's carrying the message of the genetic protein code from DNA. Messenger RNA will travel out of the nucleus through those nuclear pores, and it will go into a ribosome. And when it's inside the ribosome, this is when translation occurs, because this polypeptide, which are just links of amino acids, will be formed when a transfer RNA molecule brings them over to that ribosome. You guys might be confused. I hope you're still with me a little bit. So there's going to be other specifics of transcription and translation, but I just want you to understand the general basics of what happens. So transcription is the process of transferring the code held in DNA to a complementary base sequence of messenger RNA. There's different transcription factors which activate transcription that help this occur. This means they will separate out the DNA strand and cause an RNA to be created on that DNA strand once it's been broken. Transcription is broken down into 3 phases. Initiation is separating out the DNA strand, much like in DNA replication. Elongation is when we have an enzyme called RNA polymerase, and that will add complementary nucleotides that match up with those base pairs on the DNA strand to grow the mRNA matching sequence. So if the DNA strand, and I'm just going to type this out in the chat because there might be a question like this on your exam too. If the DNA strand that's been separated out has a base pair sequence of an adenine, a thymine, a guanine, and a uracil, and then another adenine. If this is the DNA strand, the messenger RNA strand would have base pairs that are complementary to those base pairs. So does anyone know what makes RNA different from DNA? It has one of those substitutions. Instead of one of those base pairs, it substitutes it out for something else. Am I making any sense? So I'll give you an example here. If that's the DNA strand, the mRNA strand that would create a new strand that would be complementary to that DNA strand would have the base pairs uracil, and I'll write this down first, and then I'll explain what it means. So if this is the DNA strand, take out that U in there. The complementary messenger RNA strand, the adenine would pair with a uracil, the thymine would pair with an adenine, the guanine would pair with a cytosine, and an adenine would pair with another uracil. Does that make sense? I'm going to look for some more resources to explain what I'm talking about because I think I'm confusing. Okay, it does. Okay, good. Have you guys been over this in a different biology class? Transcription, okay, thank you. Maybe a little bit in anatomy. In microbiome, perfect. Thank you. When I can't see your faces, I don't know who's lost and who's getting it. If you haven't had this before though, it's okay. All right, if you've had microbiology, yeah, I'm sure you've been over this. So again, transcription, we're creating RNA from the DNA template. Termination is just a stop signal to tell the RNA strand that there's been enough of it. So this is transcription, the steps of it: initiation, elongation, and termination. And again, what this is doing is it's taking a DNA strand, it's unwinding it, and creating a new mRNA strand off of it. We only need a single strand of mRNA because we don't need it to be double strand, but that messenger RNA will carry the message in DNA to go through the nucleus, outside of the nucleus, eventually to a ribosome. So this will be creating messenger RNA. Translation is just the step of protein synthesis where we take that messenger RNA strand and we turn it into protein. So it involves messenger RNA, a genetic code, transfer RNA and ribosomes, other translating events, and sometimes the rough endoplasmic reticulum that helps more in the protein synthesis. So the genetic code is again a 3-base sequence of DNA. We call it the triplet. On the messenger RNA, it's called a codon. There's 64 possible codons. These are the 4 bases, and there's 3 different places that the bases could go on. So that's how we get 64 possible codons. I'm just going to bypass some of this because I won't expect you guys to know all of that. But to get the genetic code, if we have a first base, a second base, and a third base and put them together, these are the codons that'll pair for a specific amino acid. So UUU on the RNA would tell the transfer RNA molecule to bring the PHE, which is phenylalanine, that amino acid, to the growing polypeptide strand. If there's a CCU codon on the messenger RNA, that'll tell transfer RNA to bring a proline to that growing amino acid chain. there's different start and stop codons as well. So when an RNA has this start codon, that tells the transfer RNA to say this is the start of the protein. And when we get to a stop codon, an mRNA, that tells the transfer RNA that, okay, we can stop bringing any more amino acids because that's the end of the protein. So transfer RNA, tRNA, will bind a specific amino acid at one end. And once the amino acid is loaded onto the transfer RNA, we call it a different name. And it basically, the transfer RNA brings this amino acid to the corresponding base pair on the messenger RNA. We call it the anticodon because it'll be complementary to the codon on the messenger RNA strand. But really simply put, transfer RNA, tRNA, will bring a complementary amino acid based on base pairs to the messenger RNA strand. And this process is similar in translation. It requires ATP, protein factors, and enzymes in an initiation, elongation, and termination sequence. This is initiation where we'll start creating the protein, the initiator tRNA, methionine. It'll detect that start codon and it'll start to read that mRNA strand and bring specific amino acids to the mRNA strand as we create a protein. Elongation involves recognizing the codon where tRNA will bind a complementary codon into the site on a ribosome. The peptide bond formation, so that it'll actually form a peptide bond between the amino acids that are added one on top of each other. And then translocation occurs when the ribosome will actually shift down 3 more base pairs on the messenger RNA. So the ribosome reads 3 base pairs at a time. And once it reads 3 base pairs, the transfer RNA puts on an amino acid and then it moves down the line to read the next 3 base pairs. And this is where elongation occurs. Once the A site is empty, a new transfer RNA will enter, bringing in its amino acid carrier. So each kind of codon will be read on the messenger RNA, and the transfer RNA will bring a specific amino acid down. There's some great videos, just probably the animations that you watch will be helpful for this. There's some also great videos on YouTube that are really kind of beautiful and shows this whole process. So these are the ribosomal subunits that'll attach onto the messenger RNA strand. And the messenger RNA strand just gets fed through this ribosome subunit. And as it's being read by the ribosome, a transfer RNA molecule brings along another amino acid. And all of these little yellow kind of circles, each one of those is an amino acid that's being formed off of reading that mRNA codon. So we've created this polypeptide, this protein that's being fed out of the ribosome as the transfer RNA brings along amino acids. Termination is when one of 3 stop codons is reached in that ribosome. Once it enters that A site on the messenger RNA, the translation stops and the amino acid will be released as the final polypeptide product. All right, questions here. So translation is the process in which genetic information that's carried on that mRNA molecule will be what we call decoded in a ribosome to form a particular polypeptide. A polypeptide means many peptides, and many peptides because peptide bonds are formed between amino acids. So another look at it, this in the lower left is showing DNA being pulled apart to form a blue messenger RNA. The mRNA will leave the nucleus and be fed through a ribosome. These are the different kind of spots in that ribosome that the mRNA is fed through. And at the P site, the 3 base pairs, the codon is read, and a tRNA brings that appropriate amino acid. And we form these little amino acids, and they travel out of the ribosome. Here's showing elongation of the amino acids being formed out. You can see here the 3 different sites in the ribosome and how the ribosome is fed through those sites. And when it's in the P site, that's where the transfer RNA brings an amino acid. How confused are we? Just put in the chat, 1, utterly confused, 10, I've learned this before and I'm an expert. And this is pretty much, and don't be afraid to say 1 or 2. It's no problem. Yeah, I'm assuming a lot of you guys maybe learned this a long time ago, or you've maybe heard about it before. I'm gonna try to find, I'm thinking of a specific video on YouTube that's made by a publisher, and I'm gonna try to put that in the modules because it's a lot nicer when you can kind of see an animation of this process than just hear me talk. Okay, so that is transcription and translation, the process by which we make different proteins. This is kind of showing where the rough ER will come into play. The rough endoplasmic reticulum will help process these proteins that are made. So what that means is the rough endoplasmic reticulum just takes the proteins that are released from the ribosome and then put them into some sort of vesicle if it needs to be transported to a different place within the cell, or if it needs to be transported to the exterior of the cell. You don't need to know the specific processes of this, but it's just talking about how the rough endoplasmic reticulum will take in the growing polypeptide or the proteins and put them into vesicles just for transport either within the cell or to the exterior of the cell. So let's just read through this summary, and then this will be kind of all we go over tonight, because I think even if you've had this before, it might be kind of a lot. I don't ask you a lot of specific questions, but I do want you to know the basics of transcription, translation, and what they mean in terms of creating proteins. So how do we get from a DNA inside the nucleus to a protein? Complementary base pairings direct the transfer of the genetic information in DNA into amino acid sequence of proteins. When we talk about complementary base pairing, that means adenine with thymine, guanine with cytosine. But if we're going to RNA, the thymine gets replaced with uracil. The DNA triplets, so 3 base pairs, are coded to messenger RNA codons. And these codons are read through a ribosome. Those are base paired with tRNA anticodons. The transfer RNA is just a type of RNA that brings the amino acid to the mRNA strand within the ribosome to start creating a protein of many amino acids put together. So here's a look at how information is transferred from the DNA to the RNA and eventually to the polypeptide. So here is a DNA sequence. And again, the T stands for thymine, A stands for adenine, C stands for cytosine, and G stands for guanine. And this just shows you how the base pairs will be paired together. So this will be an mRNA strand that is created from that DNA strand. But since it's RNA, you'll see a uracil being base paired with an adenine. Normally, if we're just replicating DNA, adenine will always be base paired with thymine, a T, but in RNA, the uracil replaces the thymine. So in all of these messenger RNA, transfer RNA, you don't see any thymines, you only see uracils. So these are the codons in the mRNA, and then the transfer RNA has what we call an anticodon. It couldn't get any more complex, right? We have codons and then anticodons on the tRNA. The anticodon follows the same complementary base pairings. When that transfer RNA reads the codon, it brings the appropriate amino acid that codes for it. If the code says AUG, that's the most common start. It's the only start code. So in the ribosome, if it reads an AUG on the messenger RNA, it says, all right, let's start making a protein. So the tRNA will bring a methionine, MET amino acid. And then it'll create the amino acid strand. And a protein is just amino acids put together. The amino acids are held together by peptide bonds. So that's why we call it a polypeptide. Poly means many. And then when we get to a stop codon on the messenger RNA strand, that tells the protein to detach from the ribosome, and it says, all right, we've made our protein, transfer RNA, we don't need you to bring any more amino acids. Other roles of DNA, DNA codes for other types of RNA, don't worry about that. Yeah. Apoptosis, autophagy, and proteasomes. This is a little important to know. Autophagy is just the self-eating of the cell itself. So cells that become obsolete or damaged basically tell their organelles to eat themselves up, and that's called autophagy. So their lysosomes basically just eat up the cell. Proteasomes disassemble these ubiquitin tagged proteins and help to recycle any extra amino acids. This would be if a damaged protein is marked for destruction, the proteasomes just recycle the amino acids within that protein to be reused. And then apoptosis, you probably have heard of this, is called programmed cell death. And this causes certain cells to neatly self-destruct. This process can begin with mitochondrial membranes leaking chemicals that activate enzymes called caspases, and they help with apoptosis. All right, let me stop the recording there.